The humid equatorial belt is the predominant region for the world's perennial plantation crops: coconuts, oil palms, rubber, bananas, cocoa and others. When planter Leslie Davidson was manager responsible for developing a large plantation in the Sabah jungle in East Malaysia, he had a problem. The problem was poor pollination. His trees, instead of providing a good crop of healthy fruit, were producing low-yielding and poorly pollinated fruit bunches. Why? Let's begin with nature's basic requirements. Good pollination of the female flower is essential if high yields of fruit are to be obtained. This is as true of the oil palm as it is of any other form of natural life. Until very recently, it was generally thought that the oil palm was wind-pollinated. But in most parts of Southeast Asia, some hand-pollination was found to be necessary to ensure adequate fruit set and reasonable yield. In wetter parts of Malaysia, New Guinea, Solomon Islands and elsewhere, each individual palm had to be manually dusted with pollen as often as every 3 days, an expensive and time-consuming business, and certainly one which was unavailable to smallholders who didn't have the spare labour or even the technical know-how. Consequently, their yields were low, and often their fruit bunches simply rotted. In its natural environment as a native of West Africa, the oil palm has evolved as part of an ecosystem which included natural pollinators. However, when introduced to Southeast Asia, the oil palm, an efficient oil-bearing plant, required artificial pollination. Why? Obviously, something was missing, but what? Instinctively, the planter didn't believe that differences in humidity or wind conditions alone were sufficient reasons to explain the need for hand pollination in Southeast Asia. Some scientists claimed that the low male-to-female ratio of the flowers was responsible. Others claimed that high rainfall in Southeast Asia washed pollen out of the atmosphere, thus reducing pollination. In 1977, Unilever arranged for independent authorities to investigate this problem. Entomologist Dr. R.A. Syed, then working for the Commonwealth Institute of Biological Control, was seconded to one of the company's plantations in Cameroon in West Africa to study pollination in the natural habitat of the oil palm. Dr. Syed is a distinguished entomologist with years of experience in tropical crops. Tests were soon able to establish that, contrary to general belief, in Africa the oil palm is insect-pollinated. There were more than 20 species of insects on the male and female inflorescences of the oil palm, and of these more than a dozen were common to both. More detailed experiments showed that at least 7 or 8 of the species played an important role in pollination. The next logical step was to go to Malaysia to examine the trees there. Neither authority, Syed or the Commonwealth Institute of Biological Control, had any commercial interest in the oil palm industry. On an oil palm estate in West Malaysia, Dr. Syed soon found a local insect species, Thrips hawaiiensis, which had become adapted to the oil palm. Thrips hawaiiensis was indeed playing an important role in pollination in West Malaysia, but proved a far less efficient pollinator than the African weevil. In Sabah, East Malaysia, the pollination problems were even greater than in the West. No insect pollinator of any importance was found. This was also true of oil palm estates in surrounding countries. Unilever had to decide whether it was worth investigating the introduction of Thrips hawaiiensis from West to East Malaysia as a pollinating agent, or was it going to be more useful to enter a second phase of study in Africa on the African weevils? The thrips answered this question for them by its general behaviour. Thrips hawaiiensis was considered unsuitable for introduction to new areas because it lives and feeds on numerous species of plants and therefore is potentially harmful to them. The Commonwealth Institute of Biological Control also believed that the thrips had acquired its pollinating role comparatively recently, whereas the Cameroon insects had almost certainly co-evolved with the palms over the millennia. Overall, the African insects looked much more promising. A second phase of study in Africa was launched, and the possibility of introducing suitable insects from Africa to Southeast Asia was growing. But which ones? From the dozen or so species regularly associated with pollination on oil palm inflorescences, Dr. Syed narrowed down the selection to 3 species of weevil of the genus Elaeidobious. One of them, Elaeidobious kamerunicus, appeared to be the most promising candidate for introduction into Malaysia and other countries having similar pollination problems. The 3-year program employed experiments which were complex and painstaking. The crucial point of these experiments was to ensure that if the weevils were to be introduced into another country to pollinate the oil palm, they would neither damage the oil palm or any other desirable plant. In order to do this, it was necessary to assess the contribution of each species to pollination, study their life histories, population dynamics, ecology, and finally to prove conclusively that they were not only specific to the oil palm, but vital to its survival. The adults of all the species of Elaeidobious feed and breed on the male inflorescences of oil palm after the flowers have opened and released pollen. They feed on the inner parts of dead male flowers. As the weevils busy themselves on male flowers, they pick up the pollen grains on the undersides of their bodies. The grains stick mainly to the thorax, abdomen, and under the wings. It was discovered that a single male kamerunicus weevil could carry up to a staggering 600 grains of pollen. In repeated experiments, kamerunicus clearly displayed the greatest searching and carrying ability. Even the industrious girls in Malaysia couldn't compete favourably with the weevil. Neither could the wind. Observations made by exposing glass slides coated with adhesive at various distances from male flowers indicated that some pollen grains blown from taller palms probably reach adjacent palms and may adequately pollinate the female flowers. But in the absence of a large number of well-placed male flowers, wind dispersal can't be expected to give regular, adequate pollination. Elaeidobious kamerunicus is the pollinator that reaches parts other pollinators cannot reach. By now, it had been firmly established that the kamerunicus weevils visited the female flowers only during their period of receptivity. A question arose. There's no food available to them on the female flowers, so why go? Dr. Syed discovered that during their period of receptivity, female flowers undergo a rise in temperature which is accompanied by an odour. This odour mimics that of the male flowers. So the weevils go to the female flowers thinking they contain food. During their visit they do no damage, they simply oblige the female flower by delivering the necessary pollen grains. Closer studies showed that the weevils are repelled at close quarters by a chemical deterrent. By this time, of course, they've deposited pollen grains. Fertilization, therefore, is achieved by deception. Scientists now had a clear understanding of how the oil palm was pollinated. The next phase was to study the weevils. Adults of the species Elaeidobious kamerunicus mate at any time of the day, usually 2 to 3 days after emergence, some even sooner. The eggs, measuring less than half a millimetre, are deposited inside the dead male flowers. The tissue surrounding them dries, hardens and shrivels, thus providing protection for the embryo inside. There are 3 larval stages, or instars. The larvae first feed on the dying tissue surrounding the egg-laying site. After they molt, they move to the base of the dead flowers, where feeding on the soft tissue is resumed. Often, the egg and first 2 larval stages of kamerunicus are complete in 2 to 3 days. The mandibles of these young larvae are weak and incapable of cutting through to the next flower. This necessitates completion of these larval instars within the same flower. Before the flower is fully consumed, the larva molts again, and at the 3rd stage continues feeding from the base of the anther tube. The larva now has stronger mandibles and is able to cut a hole near the base of the adjacent flower and resumes feeding from the base upwards. At the third stage, it can also feed on the hardened flowers. In this way, the third stage of the larva may consume 5 to 6 dead flowers. It becomes inactive for about 1 day prior to pupation. After molting to the pupal stage, the body of the larva is transformed into the adult form. The adult form can be seen through the transparent pupal skin just prior to emergence. The number of weevils found on male spikelets is related to the stage of flowering. They are few on the first day, when only a few flowers have opened, but increase on the second day and are found in maximum numbers on the third, when all the flowers have opened. Numbers then decline progressively on the fourth and fifth days, and seldom is any weevil found on the sixth day. Kamerunicus was always by far the most abundant species, even during the rainy season. The lifestyle of the weevil Elaeidobious kamerunicus was now understood. It had been established that they were able to feed and breed successfully on the oil palm. Kamerunicus next had to prove that he was specialized and specific to the oil palm, and if introduced into another country, it was important to establish that he would not endanger other plants. New tests required that Elaeidobious kamerunicus be restricted to the sequence of plants nearest related to the oil palm, Elaeis guineensis. Elaeis oleifera, the South American oil palm, is the most closely related species to guineensis. Guineensis is the principal tree used in today's oil palm industry. The male flowers of oleifera and guineensis are similar but different in their odour of anise. For this reason, it appeared that the site was less attractive to the weevil kamerunicus. Although the weevils were able to feed and lay eggs in the oleifera, the survival rates of males and females was much lower. On guineensis, their natural host tree, females of Elaeidobious kamerunicus produced a progeny some 15 times greater than females restricted to the oleifera tree. The adults reared on the oleifera tree were smaller too, indicating that the flowers on the oleifera provide less nourishment to the weevil. Although initially acceptable, in the long term the South American oil palm is not a suitable host for the kamerunicus weevils. Incredibly, they could not survive if restricted to this tree, even though it is an oil palm. This exciting discovery indicated that the weevil must be specific to the oil palm Elaeis guineensis. To be absolutely certain, scientists conducted experiments on a wider range of plants. The next and most closely related plant to be exposed to the activities of the weevils was the coconut tree. There are many varieties of coconut palm. This dwarf Malaysian variety on trial in Africa provided a convenient site to continue the tests. It was important to see how the kamerunicus weevils behaved when exposed to coconut palms. Hundreds of flowers were examined. Elsewhere, weevils were superabundant, but not a single weevil of the genus Elaeidobious was ever found. The flowers on the coconut tree are nectiferous and sweet-scented, and are visited by bees and other insects, including flies and ants, but the flowers held no attraction for the weevils. In field tests where the kamerunicus weevils were restricted to the coconut flower samples, they seemed at a loss to know what to do, and all of them dropped off within minutes. Of the larvae introduced by researchers into the male coconut flowers, none was left after 3 days, and egg-laying did not occur at all. It was concluded, therefore, that if forced to, the weevils could feed on the coconut flowers, but that they posed no danger to the crop. Many varieties of coconut palm were tested. In each case, the weevils neither bred nor fed satisfactorily, if at all. Flowers of about 50 other species of plants were tested in the laboratory. None of these plants could keep the weevils alive for any length of time, and no eggs were laid on any of the flowers. In field tests, no single kamerunicus adult was ever attracted to these plants, but as they always were to the flowers of the oil palm, Elaeis guineensis. Elaeidobious kamerunicus became the most heavily researched insect to be considered for crossing a national boundary. The 3-year research program started by Unilever and the Commonwealth Institute of Biological Control was at last finished. The results showed that the weevil kamerunicus was highly specialized and completely specific to the oil palm and was unable to feed or breed on any other plant species. Studies had firmly established that the release of kamerunicus into a new environment would not only be harmless to other plants, but would be a step towards improving the ecological balance in oil palm plantations by reuniting the palm and the weevils which had evolved in mutual dependence in Africa over the millennia. A perfect example of symbiosis in nature. The weevils were brought from Cameroon to England, where they were re-examined by Unilever and the CIBC, repacked, then flown to Kuala Lumpur. The Malaysian Federal Department of Agriculture, who had monitored the African research throughout, accepted them and after their own series of tests allowed the weevils to be released on the 21st of February 1981 on Unilever's plantation in Kluang, West Malaysia. Within a month, weevils were also released in Sabah, East Malaysia. Three months later, after similar tests to reassure local entomologists that kamerunicus was specific to the oil palm, the weevils were released in Papua New Guinea and later in the Solomon Islands, Indonesia, Thailand, Burma, the Philippines and some countries of South America. Southeast Asia is now taking advantage of an efficient pollinating agent produced in Africa by nature after millions of years of trial and error. Within months, improvements in fruit set were observed. The effects of improved pollination were dramatic. After the first full year, yields of fruit increased by up to 50%. The Malaysian Minister of Primary Industries, Paul Lee Yong, estimated that as a direct result of the introduction of the weevils, in 1982, Malaysian plantations yielded an additional 400,000 tons of palm oil and 300,000 tons of palm kernel, with a total value of over 370 million Malaysian dollars. The benefits will be particularly marked for smallholders. Yields from small holdings in Sabah have increased by nearly 40% since the introduction of the weevils, and at no cost to the farmers. Eliminating the need for hand-pollination, which was imperfect anyway, has helped to resolve a developing labour shortage. The large numbers of pollinators formerly needed have been released for more productive work elsewhere on the plantations, and no redundancies have been caused as a result of the weevils' arrival. The introduction of the weevils and the ensuing benefits to the Malaysian economy and the economies of neighbouring states are the direct result of the initiative of Unilever's plantations management and its facility to transfer know-how effectively within an international business. The results of this research programme have been described as one of the most important biological innovations, not only for tropical agriculture, but also for third world countries and their future. And others.